So ACR, you ask, how does the mutation on the gene affect the production of the protein? Okay, that's the super good question and the very basis to understand what a mutation is, okay, and why mutations give symptoms, basically. Okay, so we have a gene, or everything that is alive, basically, has a gene called ZC4H2. The ZC4H2 gene wasn't discovered until 2013 by Dr. Vera Kalschuwer, which is one of our advisors, by the way. So we didn't know that it existed and didn't know what it does. We still don't know what it does, so that's what we try to elucidate. So genes are, excuse me if I'm going to be very basic. If you think that this is not of interest, just let me know, okay, and I will advance. So this gene appears to be incredibly important for the formation of the nervous system, of the central and peripheral nervous system. We don't know exactly what it does, but we start having an idea. So a gene, basically, which is contained in your DNA, you know, the DNA is like organized in functional units called genes. This gene produces a protein. So genes basically produce protein. They are the codes to produce proteins, right? And the ZC4H2 produces a protein called the ZC4H2 protein that is very important. Does a lot of important things for life. One of those important things that it does is that, you see, proteins can be, to understand a little bit, proteins can be structural proteins, like your muscles, like your tissue. Proteins can be enzymes, proteins can be hormones, proteins can be receptors on your cells. Proteins can be a lot of different things, but this particular protein appears to be a boss protein, meaning that it gives DNA the signal to produce other proteins. So it doesn't specifically has a function like an enzyme or a hormone or a structural protein of our body, but it goes and sits on DNA or on RNA, and it gives the cell the signal to modify or to produce or stop producing other proteins. Now, that little bit is that we're investigating now. So basically, that's a problem, because it means that we call it transcription factor. A transcription factor is this incredibly important proteins that command order and are the bosses of other genes. So it is very high up in the development. So that protein is produced very early in the development, and it starts commanding the function of many, many, many other proteins in the body. Okay, so that's the basic. Please hand up if I'm too fast or you don't get something. So this protein, ZC4H2, produced by the body, is vital. It's not only important, it's vital for life. Why do we know that it's vital for life? Because we have animal models in which we go and take out the ZC4H2 protein, we call it a knockout, and the respective animal dies. So the mouse or the zebrafish or the anything that they have done with animal models die. They are not viable for life. This protein, ZC4H2, is found only in embryos, and not only, it's found mostly in embryos, in tissue of embryos, and in adults is found but reduced in reduced quantities. That means that the ZC4H2 protein has a very, very important function early in life, in the development, right? So basically, when the protein didn't form correctly because there was a mutation on the respective gene or the code that produce the protein cannot work. So what are mutations? So imagine that you have your DNA that is a code, or 4 letters, C, T, A, and G, a very, very long code of hundreds of thousands of those letters. In the case of ZC4H2 gene, there are hundreds of thousands of those letters. One letter goes wrong. or a piece of the gene gets, or God knows why, is deleted. So the protein that should have been produced based on this code is either not produced at all, or it comes with a defect. Imagine you have these are proteins. Imagine that proteins look like, proteins look like this, basically, but for the sake of this explanation, they look like pearl necklaces, right? And every pearl is an amino acid, right? Proteins are formed by amino acids. And the amino acids, the sequence of amino acids is incredibly well established and cannot change in nature. Cannot change. When this necklace of pearls, you know, they're all beautifully pink, all of them, one of them is not there, or one of them is black, or one of them, you know, it's a little bit not round. This whole protein cannot function correctly. So it doesn't fold beautifully and it doesn't function, but it becomes like, and doesn't work. We call it a truncated protein, truncated or incorrect protein. That in case that one amino acid was changed. In case the whole piece of gene was deleted, the protein is probably not transcribed, it's not formed, so your body doesn't produce this protein. If your child was not able to produce, a male, I'm talking about males, I will explain later well, was not able to produce this protein, that child will never be born. It's not compatible with life. If that ZC4H2 protein was a little bit truncated or misformed, let's say, because one of those amino acids changed, it can be a very good functional protein, and then you have a child that doesn't have symptoms or has a very mild symptoms, or it is a protein that has a big defect and it doesn't work properly, and then you have severe symptoms. And that is, you know, how the mutation affects a protein. Now, do we know how a mutation actually affects a protein? It depends which part of the gene was mutated. That is what it is so important to know the variant. When we say variant, we refer to the type of mutation that your child has. And that's why we always ask, you know, give me the variant, because the variant will tell you exactly where in the gene the mutation happens. And that gives us an idea or how the protein, if the protein was, you know, became short, long, bad, or almost not bad. So depending on where the mutation happened and the type of mutation, you can predict how the protein was formed. And again, if that protein is completely unfunctional, then you have a big problem and the protein still works a little bit, then you don't have much of a problem. How do we know that? Those are studies that they need to see if your child produces a protein and the protein is functional, but they are incredibly difficult to do. I'm not aware of any of our children that have had those functional protein studies. They have only been done for a few variants in animal study. And it doesn't matter at the end of the day, it does matter a lot for research, but for us on a day-to-day basis, what we look is at the symptoms, right? Symptomatically, you can predict, okay, the ZC4H2 protein in this child is so changed that there's a lot of consequences. Or this child is so mild that probably the ZC4H2 protein is still functional.